And this is something that's fundamentally really difficult because it is sort of a moving target. Also, when you have a big tree with lots of species, there are so many different possible trees, as we know, that you can't really look at all of them. It's just not possible. So you have to you have to sort of look around here and there within the different possible kinds of trees, and then try to find the best tree. Searching with a search algorithm, basically. But then for the whole tree is like one big hypothesis, and then each little clade is really its own hypothesis. So there's the whole tree here, and then there's a hypothesis about clade A here, and there's another hypothesis about clade B, and what the relations are within that clade. So what we can do is we can look at these individual little branches or clades and see how much support there is for each individual part of the tree. So one way that we can do that is this bootstrapping approach. You don't need to worry about the details of this, but essentially what you can do is, especially when you have lots and lots of data, you can take a random sample from your data set, random sample of traits, characters, could be base pairs, could be morphological characters, and then you build a tree based on that. And then you find the best, the most, either the most parsimonious or the most, the tree with the highest maximum likelihood. You evaluate all the trees based on those data, and then you resample the data again. You take another random sample of your data and you build another tree, or another bunch of trees, and you find the best one. So what this does is it allows you to ask how sensitive is the tree, is that best tree? How sensitive is it to particular parts of your data set? So again, you take the full data set, you sample some of the traits with replacements, you give it a twice, and then this gives you a whole other way of looking at to test your hypothesis. So when you do this, you can take your character data, you build a tree, you sample it, you build lots and lots of trees, lots and lots of best trees based on those data, and then essentially what we can do is we can report the confidence interval, or how confident we are in particular clades. So in this case, we're 90% of the time, or 90% of our bootstrap samples from our data, we found that the best tree always had A1, A2, and A3 together, 90% of the time. So that gives us a way to say, what's the likelihood that the A's are really a clade, that they really came from a common ancestor. Should I say that again? Okay. So we'll see this a lot of notation that uses some form of confidence interval. And so one example is testing hypotheses about mammalian evolution. So the Theria hypothesis posits that the mammals are most closely related to marsupials, and the monotreme, platypus, and whatnot are the outgroup of that. Okay? Alternative hypothesis is that the placentals are a sister taxa to the taxon that includes both marsupials and monotremes. Okay? So the question is where do we fit in terms of so here's a tree. So these are substitutions per site. So this is one substitutions per base pair, right? The length of these tells you how much evolution is happening with the last ancestral node. And so that means that the chicken is really different from this whole crowd over here. It's evolved a lot in the meantime. In contrast, the platypus and the echidna have evolved only a tiny bit since they diverged. the numbers tell us in those bootstrap samples how often the individuals in this clade were in the clade, right? So 100% of the time, for all of these samples, these things were all together, right? In contrast, for all of those resamples, the bat and the hedgehog only wound up as sister taxa 50%. So 50% of the time, it's like flipping a coin, so we don't really know whether the bat and the hedgehog are the closest relatives or not, but we can be quite sure that the marsupials are sister taxa to the placental mammals, and that the monotremes are outside that. So we have 100% work here and here, and that tells us that relationship is something here. So it's important to recognize that when you look at a tree, the whole tree is one big hypothesis, but there are individual hypotheses embedded within it. And fortunately, we can evaluate those hypotheses independently. So, given data like this, there probably won't be a lot of graduate students out there trying to retest this hypothesis, right? Whether the placental are sister to the marsupial versus the monotreme. Yet, there probably are grad students out there trying to figure out how the bats and the hedgehogs are related to each other, and how they're related to the pig and the cow. At least we hope so. Okay. Questions? All right. Okay. So, just a couple more little tidbits about trees. So, once we have trees, we can use them to ask other kinds of questions. So, for instance, we might be interested in knowing whether there's a relationship ecologically between arboreality, right? So whether things live in trees or on the ground, and whether those same things are nocturnal versus diurnal, right? So we have 10 species here, and we never found any that were diurnal and arboreal, nor did we find any that were terrestrial and nocturnal. So if you run a simple test, a simple stats test called the chi-square test, basically evaluates table data like this. And so what would we would expect if we just threw 10 species at this table is that 2.5 species would wind up in each bin, right? But in fact, that's not what we find. We find this very distinct pattern. So the likelihood of finding this pattern is very low, right? So from a traditional standpoint, we would conclude what? You think that arboreal species are able to be diurnal? Or is it just something that can't happen? Do you think there's a slight pattern here? Yeah. Okay. Right. So that's the idea here, right? So things that live on the ground, if they're out there in the daylight, they can eat really fast, right? And they're, I got the other around. That things that are hiding in trees are out and about when it's nighttime, when they hide. So apparently there's a strong correlation. Now, once you have trees, though, you can ask these questions differently. So if we have, imagine that we have these data here, right? These are our 10 different species. 4 of them are nocturnal and arboreal, and the rest of them are all diurnal and terrestrial, right? So imagine that we have a tree here that's not based on our observations about these creatures. Imagine that we have this tree that's based on DNA, for instance, has nothing to do with their characteristics. And imagine that we find this, that we find that there's a clade that supports these 4 species being related. And according to this, the mapping of these traits onto this tree, then it certainly looks like diurnality and arboreal lifestyle evolved once and evolved together. that relationship? Are you guys convinced that it has to be that way? Because really, what this tells us is that we don't really have 10 species to compare, right? We have one. We have one clade where this happened and it happened together. And the odds of it happening together, or the odds of, put it another way, the odds of one of these happening at the same place as the other one happened is much lower than 1 in 10. And so when you take this kind of approach, you can ask questions much more carefully. You can ask them independent of the phylogeny. And I'm going to talk just a little bit about that. Another example of where this can be important. So for instance, here we have a figure that shows pesticides. Well, age of first reproduction as a function of pesticides, and as you can see, according to this figure, individuals or species with larger testes are older when they first reproduce. Okay? But what if instead we actually have two clades that are quite different from each other, and then within the clades we can see that in fact there's a negative relationship with this clade and a negative relationship with this clade. So by simply taking a bunch of species and assuming that they are independent replicates or independent experiments and treating them that way, you can trick yourself. But once you know the phylogeny, then you can ask these questions in a much more robust way. So this idea is that if you have some tree, you can ask questions about whether A and B differ in some variables. So are they correlated in their nocturnality versus their arboreality? And you can ask whether these are similar, and then you can ask whether this parent is similar to C, and then you can ask whether this parent is similar to D, and then you can ask whether D and E are different from each other. And so by asking the question in a nested fashion within the tree, you can test a hypothesis that's robust. Because basically each of these divergences gives you a chance to test the hypothesis. But simply looking at all the species and lumping them together doesn't give you that power. You can trick yourself with it. Okay. So one example is whether fire-adapted traits and fire-promoting traits have evolved together and at the same time. So here we have a pine tree looking out west, has protective bark and the ability to resprout, and cones that open when they're burned, right? So those are all fire-adapted traits. On the other hand, they have lots of tiny needles and twigs that are highly flammable, and they keep their dead branches down low, and these things promote fire. So the question is, have these things evolved together or not? And we can look at that by looking at a phylogeny, by asking whether these different species that are clades within a tree, within a phylogenetic tree, not a pine tree, whether they live in phyloprone environments or not, right? So here, we have all of these species represented by some common ancestor and some other common ancestor here, where we can infer that the common, because most of these are not in fire-prone environments, non-fire environments, we can infer that the ancestor was also a non-fire environment, and therefore we can compare our recreated traits, basically, if we go back in time, the ancestral traits here versus here, and ask, were they evolved together? We can do the same thing here, and we can do the same thing here. But the trick is, even though we have all these species, a whole bunch of species that are in a fire-prone environment, they're all related to each other, and therefore we can't treat them as being independent casts of the hypothesis. And the answer is, in fact, with very, very few opportunities to actually test it, we can't really know The answer is well. Okay, now I just want to mention very briefly the end of chapter 2 talks about gene trees. I'm not going to tell you about gene trees much, or expect to know much about them, except that for the simple fact that populations and species may evolve independently from the genes that they contain. And basically what we can see here is we have 4 species of organism. I don't recall what they are. But within them, all of these little lines are a whole bunch of gene trees that are superimposed over it. And what you can see, what you might be able to see, is that some of these divergences, like here, this green gene diverged way back here, and yet those lineages wound up in different species. And offset. All right, now let's look a little bit. Questions for you on gene trees? Basically, they can evolve different species. Okay. The great thing about a textbook is that there's really more in it than we need to know. So you don't have to know everything. Let's get some parts. All right. So in chapter 3, Katona lays out a bunch of evidence for evolution, and I'm just going to skim through some of that. So the evidence that he suggested is important for this whole, for the big evolution, whether it's real or not. And I'm going to run through this here and then just give you a few examples of each. And some of these you've seen already. Hierarchical organization of biological diversity, homology and synapomorphy, embryological similarities, developmental similarities, essential characters, the characters that are no longer useful to an organism, convergence, when different evolution pathways lead to the same place, core design, geography can tell us a lot about showing us whether supporting phylogeny or vice versa. I'll talk about that a little bit more. And then there are a lot of intermediate forms, so lots of things that clearly show very clear gradations between different forms, which suggests that they're intermediates along some evolutionary spectrum. And of course, similarly, there's lots of microevolution that we can observe on the not necessarily daily basis, but over the course of months, you can watch those things evolve. Okay, so hierarchical organization, as Linnaeus observed, and he did a mighty good job of it. You can lump different organisms together, related organisms, and they have a hierarchical display of their characteristics. So feathers evolve here, all of those have feathers. Tetrapod limbs evolve here, all tetrapods have 4 limbs. For the most part, we don't find that feathers are outside of birds. We don't find that tetrapod limbs are outside of birds, outside of tetrapods. Homology and also synapomorphy. So homology being characters that are descended from a common ancestor, synapomorphy being shared derived characters. These are simply showing us various bones that you have in your own body. So in the arm and the forearm, your fingers, all of these are found in lots of different organisms. They've been adapted for grasping in humans, adapted for swimming, adapted for running, and adapted for flying. So the same bones, used for different things. Which suggests that they have a shared evolution. Similarly, various parts of leaves, in particular plants rather than stipules, have evolved to have different forms. So here they've evolved into tendrils. Here we have leaves that are modified into tendrils. Here we have petioles that are modified into tendrils. So in fact, this is a figure showing convergent evolution, not homology. I will the slides and we put them in. So, yeah, so these are different ways that climbing has evolved in plants in a convergent way, not on all of them. Did you catch that? Okay. So here we have another example. So homologs are morphological characteristics that are that have a shared common ancestors. In genes, we also have homologs, and we have two different ways of looking at them. So here's another typo here. That should say ortholog, not ortholog. Anyway, so if we have some ancestral species here with some gene, the gene could, first of all, be passed down here, right? And then one version of the alpha gene winds up in species 2, another version winds up in species 1, and the red arrow here tells us that those are orthologs, right? Now orthologs mean that the gene itself in two different species has a common ancestor. Okay? In contrast, paralogs are genes that have been duplicated. So this gene is duplicated here. Now we have two different flavors of it, two different copies of it, and I don't mean two copies on two different chromosomes. I mean another whole copy of it. So two copies here. They both get passed down. So now these are paralogs, duplicate versions of the same gene. We see this in lots and lots of instances. And some examples are hemoglobin. So hemoglobin has basically duplicated itself a bunch of different times. So in the placental mammals, we have several copies of it. And they do different things. And the Hox genes as well. So in this case, all of these organisms have at least one copy of the Hox gene. So in that case, between species, right? So this Hox gene and this Hox gene in tunicates versus Amphioxus, these are orthologs. Within a particular species, the lamprey has 4 copies of it. Those are paralogs. Okay? We'll talk about these a little bit more when we get to evolution and development. So Hox genes in particular are really important for development, segmentation, and things like that. Okay. So organisms have embryological similarities. So here we have human, possum, chicken, and salamander. As organisms develop, they typically gain more and more differentiation. When you go further back, you find that they are more similar. And so that's another form of evidence that we share a common ancestor. Another good example of residual characteristics. So lots of organisms have evolved sort of in caves, in places where there's no light, and they all have some rudimentary evidence of their eyes. So eyes don't do them anything for them anymore, and yet they still exist. They still have some form of their eyes. Similarly, humans have an appendix. Residual character that doesn't move before us. In fact, it's harmful. Convergence is another good example. So, and I should have my picture of those leaves here in our climbing plants. But the vertebrate eye and the octopus eye evolved completely independently, and yet they do very similar things. So an example of convergence is this eye. There are lots of other examples as well. Suboptimal design is another bit of evidence for evolution. In this case, the vertebrate eye has a blind spot in it back here. So it's just due to the design. So in this case, vertebrate eyes are designed poorly compared to the cephalopod eye. a little bit about geographic distributions and how they support evidence for evolution. So Africa and South America used to be stuck together. You go way back in time. Tanzania and then Gondwanaland, Africa and South America were stuck together here. They eventually split. And we can see evidence for this in the evolutionary history of the species that lived on these continents. In particular, the rat-type birds are distributed across this area here. These continents used to be stuck together a long time ago. And they're flightless, right? So they don't cross water very well. And it turns out that their phylogeny reflects the way that Gondwanaland broke up. So if we look here, the emu and the cassowary, right, are sister taxa, which suggests that New Guinea and Australia most recently split off. You go further back in time, the Kiwi and New Zealand split off from that. And a little further back, the ostrich split off from these continents with, because it was riding a lot on Africa. So, and of course, the whole split here splits up all these, the South American species from Koreas from all of these. So the take-home message here is what we know from geologic evidence, the order in which the continents split up, and in fact, the evolutionary history matches that very well. So this is a good question.
Can you explain a little bit more convergence, like compared with like the flowers before?
Yeah.
So isn't there like
convergence.
different types? Like isn't there like parallel and then like reversal convergence? Doesn't that fall under the same thing?
Yeah. Well, homoplases. So there are lots of things that can appear as homoplases. Right? These are things that look the same, but not for the reasons that we think that they are. Right? So having an object in humans and cephalopods is a homoplace. We both have vision, but it's not because it's a shared derived. I believe in the first lecture or the second lecture I showed an example of organisms that live in brightly colored dark colored habitats. Remember the pocket mice were brown or light tan color when they lived in sand. They were black when they lived in lava fields. That's another example of convergent evolution. Okay. So intermediate forms, more evidence. So evolution rarely jumps all the way from this organism all the way to this organism. In fact, we find that there's a lot of intermediate forms in between. And on the microevolution end of things, we've talked about this already, but here we have time on the x-axis in red. On the y-axis we have in blue, and on this y-axis we have the amount of drug use here and the resistance to that drug in red. So you can see that they essentially follow each other as the drug is used more, you get more evolved resistance to it. So over fairly short time scales, you can see evolution of resistance to lots and lots of different antibiotics, and in fact, getting to the point where we're running out of different ways to kill bacteria. Okay, that's what I have for today. So next time, read chapter 4.